Let's continue on with Unit 7, the Rh blood group system. This is Part 2. Now that we've learned about the different Rh antigens and the Fisher-Reyes and Wiener nomenclatures, let's talk about phenotyping. There are 5 reagent antisera available, and I'm talking about routine antisera. There are other special typing seras for the special Rh antigens, but we're going to talk about the ones that are most frequently used. Only anti-D is required for routine testing. The other typing sera are used for typing red cells to either resolve antibody problems or conduct family studies, with resolution of antibody problems being number 1. The agglutination reactions, positive and negative, will represent the phenotype. There is no anti-little d because little d is an amorph with no genetic product. Then we use statistical probability to determine the most probable genotype. Let's revisit phenotype versus genotype, because you will be given an Rh phenotype and you must be able to determine all potential genotypes. And I'll try and give you some tips and tricks to make it a little bit easier. As you recall from the genetics lecture, the phenotype are the actual antigens detected on the red cell using special typing sera. Has nothing to do with genetics per se, it's simply the antigen is present or absent. The genotype is the actual genetic makeup of an individual, which can only be predicted using the phenotype and by considering the race or ethnicity of an individual. Only family studies can determine the true genotype. So here's an example, which I will do a more lengthy discussion of how this is done on the next slide, but this is just a quick and dirty explanation. So here's the phenotype. It's a big D, big C, big E, little e. So the big C is homozygous. How do I know that? There's no little c. So they had to have gotten a big C from mom, a big C from dad. That's why this antigen was the only one detected on the cell. For E, the patient is heterozygous because there's big E on one side and little e on the other. They inherited both antigens. When you're determining the genotype, you must always, if a person's D positive, think about them as being homozygous for D and heterozygous for D. Because remember, we don't have any anti-little d. We can only assume through family studies or doing the phenotyping what a patient's genotype may be. So for the first, we'll look at the D, we'll put it on both sides. The slash indicates a gene complex inherited from mom and the other one from dad. So we have a homozygous D, or whenever a person's D positive, you must always consider them as heterozygous. So we'll have a big D on one side, little d on the other. And then we consider the C, and we just plop it in because they're homozygous. So they're going to have big C on both sides of the equation. Then when we look at the big E typing, they're heterozygous. So look at the first example. We put big E on one side, a little e on the other. That's the only combination that we can make for a homozygous big D. Now you could flip it. You could put the big D, big C, little e on the left-hand side. That's not going to change the genetic makeup. Whether they're RZ-R1 or R1-RZ, it's the same genetic makeup. So that's done. Homozygous is done. Then when we look at the heterozygous D, we have to consider, again, you put the big C on both sides for each because they're homozygous. But for the E blood group system, you'll put big E on one side and little e on the other side, and then you look, and if you flip the D's, that makes a whole new genotype. So on the one, we have an RZ little r prime, and on the last one, we have little ry r1. So for this phenotype, these are all the possible common genotypes. Determining genotypes, Rh genotypes, is usually a struggle for many of my students, so I want to slow this down and just take it step by step by step in converting a phenotype into a genotype. So again, we have the phenotype for this individual. They're big D, big C, little c, so they're heterozygous for C. And they're little e positive. So that means they're homozygous for little e. How do I know that? There's no big E present. When we added the drops of anti-big E to the cell, there was no reaction. So take this step by step by step. For the possible genotypes, again, I can't emphasize enough that for D positive individuals, you must look at the individual as homozygous for D and heterozygous for D. So we write D/D or D/little d. The next antigen we're going to look at is the C. This individual is heterozygous for C, as the patient is positive for both antigens, big C and little c. So let's fill those in on both sides of the equation. For our homozygous, we have big C, little c on opposite sides, and whether we flip them, it doesn't matter because it would be the same. But if you look at the heterozygous D type, this could result into different D types. So we have big D, big C, little c, or when we flip, we would have big D, little c, and little d, big C. There are 3 possible genotypes. So spend a little time looking at this and figuring out why we can have 3 possible genotypes. The individual is homozygous for little e, so you just fill those in. Plop, 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 little e on both sides, and we're done. So there are 3 possible genotypes from this phenotype. Now, you must convert the genotype to Fisher-Ray shorthand. So remember, big D, big C, the D is a big R, and the big C is 1. The big D on the right-hand side is big R, and there's no uppercase, so it's a big fat 0. So R1, R0. Then we have big D, big C, which is R1, and then all small, which is little r, all by its lonesome. And then the last one is big D, little c, little e, which is R0, and then little d, big C, little e, that's a little r prime, because the big C is positive, the big C is present. Then eventually with time, you will learn the most probable genotypes for the most common genotypes. And if this person is white, their most probable genotype is R1 little r. If they're black, they're R1, R0. So knowing the ethnicity allows you to come to a more accurate conclusion as to the actual genotype of this person. Genotype frequencies, you'll always refer to your textbook or whatever resources you have available in your laboratory. Again, with practice, it just becomes almost second nature. I can look at a phenotype and immediately do the genotype in my head and recognize the most common. But that's because I have over 30 years experience. Genotypes are always listed as presumptive or most probable, because usually you're dealing with one patient, you don't have access to their family. So again, presumptive or most probable. Genotypes will vary in frequency in different racial groups. For example, RO is very frequently found in the black population. So when determining the most probable genotype for big D, little c, little e, race becomes important. So I've given you examples of the gene complex, the shorthand, and statistics. Statistics are something that is very, very important in blood bank when you're looking at the frequency of either a single gene antigen or a gene complex. Molecular testing is becoming more popular, but is still too expensive for individual transfusion services to do it. It's becoming more popular in the donor centers. You cannot use anti-sera on recently transfused individuals, so molecular testing can help differentiate. For example, if you're doing a microarray, you can have the different probes for the different complexes. So you can determine the genotype of the transfused person as well as the person who's been transfused. Anti-sera are not available for some antigens. So molecular testing has been being developed for all blood group genes. D zygosity can be determined using genotyping. Fetal genotyping for D can be done on fetal DNA present in maternal plasma. And this is kind of a game changer. We can determine early on in a woman's pregnancy, especially if she's already sensitized to the D antigen, whether or not her fetus is D positive or D negative. Monoclonal reagents from different manufacturers react differently with variant D antigens, and molecular test is specific. So if you remember molecular, we're taking this specific piece of DNA that codes for that specific gene, and we're then able to detect it. And with multiplex, again, especially in donor centers, to be able to screen donor units for the most common antigens that patients make antibodies to would be incredible. I attended a workshop about a transfusion service where they pulled open a drawer and it was not only labeled A, B, O, D pos, D neg, it also had Cal neg, Big E neg, little C neg. I mean, it was amazing. It was a blood banker's dream, to where if I have a problem patient, I could just go to the drawer and select that unit which matched my patient and had it be negative for those antigens they had antibodies to. So for right now, typing sera continues to be the gold standard, but I guarantee you this will change in the future as the technology improves and the prices come down. What are some uses of Rh phenotyping? Parentage testing. So if you have a mom who's homozygous for C and a dad who's homozygous for C, and you have a child who's heterozygous, that would be a direct exclusion because the child inherited an antigen for which both the mom and the dad are negative. Predicting hemolytic disease of the fetus and newborn. So again, this mainly involves typing the mom and typing the dad, because if mom is D negative and dad is homozygous for D, then the baby will inherit the D antigen. Confirmation of Rh antibody specificity. Once we identify an antibody in a patient's serum, how you confirm it is you make sure the patient is negative for that antigen. For example, let's say that you've identified an anti-Big C in the patient's serum, and you go back and test the patient, and they're Big C positive. You've made a mistake. Something's wrong. Either you misinterpreted your panel, or maybe you got your cell suspensions from different patients mixed up. So again, it helps to confirm the identity of an Rh antibody. Locating compatible blood for recipients with Rh antibodies. Once you identify an immune antibody in a patient's serum, you've added hundreds, if not thousands of dollars to their transfusion bill, because for the rest of their life, they must get antigen-negative blood. So if a patient has anti-Big E, you need to type the donors and make sure that they're Big E negative as well as cross-match compatible. And we'll talk about that more when we get into the identification of antibodies. What is the protocol for Rh phenotyping? You mix your unknown, whether it's your donor cells or your patient cells, your unknown cells with known Rh antisera and mix it up. It's just like an ABO type. Now you have to follow the manufacturer's instructions. Agglutination indicates the presence of the antigen on the cell and determines the phenotype. Use published frequencies and subject information to determine the genotype. Again, once you have the phenotype, determine all the possible genotypes, then look up in your reference as to what is the most probable genotype for your patient. So again, the D antigen is the most immunogenic in the Rh system, meaning if you are D negative and you're exposed to the D antigen, the chances of you making an immune anti-D are 80% or higher. D negative individuals should always receive D negative blood. D negative individuals receiving D positive blood may produce anti-D. What I liked about this slide is it shows, according to genotype, which genotypes have the most D antigens to the fewest D antigens. And this will come into importance later on when we're talking about mothers and babies. Let's talk about another topic that confuses students, the weak expression of D. Not all D-positive cells react equally well with anti-D, and weak expression of the D antigens occurs more frequently in the black population. Red cells that are not immediately agglutinated by anti-D must be tested for the weak D by the indirect antiglobulin test method, and you should have already done this a couple of times. What happens is when you determine that a patient on immediate spin is D negative, then you perform a weak D test. You incubate the cells with anti-D. You incubate at 37 because that's body temperature. These are IgG antibodies. That's where they like to react. During the incubation, the coding or sensitization of the D antigens will occur if the D antigen is present. In other words, the reagent anti-D will attach and bind to the weak D antigens on the cells. But you can't see it because they're IgG. So what we do is we wash 3 times to remove all the contaminating proteins, and you add anti-human globulin serum. If IgG from the reagent anti-D has attached to the cells, the AHG will bind to the anti-D coating the cells if it's present. If the weak D test is negative, the individual is truly D negative. If the weak D test is positive, the individual is D positive weak, and we record it as D pos superscript W for the interpretation. You must memorize the 3 mechanisms for weak D, which are genetic, position effect, and mosaic. These result in differences from normal D expression. It could be a quantitative difference that perhaps you've inherited a weak D, or it may be due to a position effect, or it could be qualitative, an actual difference in the D gene itself. For example, mosaic D. And this particular weak D can produce anti-D. I want to say that one more time. You have genetic and position effect, which have to do with a quantitative effect on D expression. The mosaic D, it's actually a qualitative difference, and these individuals could potentially produce anti-D. In the genetic weak D, this involves inheritance of D genes from your parents, which results in lower densities of D antigens on the red cell membranes. So it codes for less D antigen, and it may be to the extent that you get a weak positive or negative on immediate spin. So this graphic illustrates the differences between a genetic weak D and a normal D person. So on the left you see an RBC with normal amounts of D antigen, and on the right hand side you see a person who has very spotty D antigens, which will result in a weaker expression of the D antigen to the extent that on immediate spin it is negative. One thing that I want to reiterate, that when you're doing D typing, you should always get 3+ to 4+ reactions. A weakened expression of the D antigen could be due to position effect, and this is called C transposition effect. The D gene is in trans to the C gene. They're on opposite sides. For example, big D, little e, / little d, big C, little e. Notice the D and the C are on opposite sides, which could lead to steric hindrance, which results in a weakening or suppression of expression of the D antigen. So we have a weakened expression, which again, on immediate spin, you may get a negative result. So here's an illustration. Once again, C in the transposition to D, we have the weak D. But if C is in the cis position to D, which is the lower illustration, it is not weak D. You'll have a normal expression of the D antigen. Partial D is absent of a portion or portions of the total material that comprises the D antigen. It's known as partial D. The old term was D mosaic. If a partial D patient is transfused with D positive red cells, they may develop an anti-D alloantibody to the part of the antigen, the specific epitope that they are missing. So let's look at the red cell on the left. We see a full complement of D. The entire D antigen is present on the red cell. If you look at the picture on the right, this individual is missing a piece of the D antigen. And if they are transfused with normal D positive blood, they may make an antibody against the piece they are missing, but it looks just like an anti-D. You need to totally understand the significance of the weak D as a donor and the significance of a weak D as a patient. Donors, if they are weak D positive, they're labeled as D positive. The weak D antigen is substantially less immunogenic than normal D antigens, but it has been known to cause severe hemolytic transfusion reactions in patients with anti-D. So again, if you are a weak D positive person, as a donor, you are considered D positive. Let's look at patients. If the weak D is due to partial D, they can make an antibody to the portion of the antigen they lack. But how are we going to determine this? You can't. You can't. All weak Ds, they're negative at immediate spin and they're positive at Coombs. So how are you going to know whether it's due to a position effect or it's due to the partial D or if it's due to genetic expression? You don't. If the weak D is due to suppression or genetic expression, theoretically they could give D positive. How are you going to prove it? It's going to be very, very difficult. So standard practice is to transfuse based on the immediate spin reaction with anti-D. I want to say that one more time. How you're going to select your D type for your patient is based on the immediate spin reaction with anti-D. So on immediate spin, you get negative, and then you do the weak D test and it's positive. It matters not. You're going to transfuse with D negative. A lot of transfusion services don't test for weak D, except for certain circumstances. A lot of transfusion services will do a baseline, meaning when we first see you, we will do a weak D test. But regardless of what the outcome is, we're still going to transfuse you with D negative blood if you're a weak D. I always like to know what the weak D type of my patient is. Weak D testing on donors by the transfusion service is not required because we have labeled that unit D positive. It won't matter. There's no way it's going to be transfused into a D negative person. So it doesn't matter. The transfusion service, we do not retype D positive donors because it doesn't matter. Again, weak D testing on patients is not required except in certain circumstances. This is the end of part 2.